Hello, I am going to show you how to do a yeast mutagenesis and serial dilution experiment. The purpose of this experiment is to see how UV radiation affects the mutation rate and survivability of cells. We start with the serial dilution, where we resuspend yeast cells before plating them and giving them varying durations of UV exposure. We dilute the yeast sample from a toothpick or pipette tip because it contains millions of cells and we need to be able to count resulting colonies for this. We are going to start by grabbing 5 test tubes and labeling them 10 to the 7th, 10 to the 6th, 10 to the 5th, 10 to the 4th, and 10 to the 3rd. Here I have my 1000 microliter micropipette, and we are going to set the value of volume intake to 900 microliters. We transfer 900 microliters of deionized water to each test tube. When we do this, we use a new P1000 pipette tip each time for contamination reasons. Here I have a culture with HB1 yeast, which has a mutation in the AD1 gene that makes it pink. In our mutagenesis experiment, we want to see how many colonies mutagenize to have a white phenotype. We are going to take our P200 micropipette and set the volume intake to 100 microliters. You can see that I am using a pipette tip to gently poke a yeast colony. There is some pink stuff on the micropipette, that's the yeast. I transfer that to the 10 to the 7th test tube and resuspend the yeast cells by flicking at the tube. Then, we do our mixing technique with the pipette, where we push and pull solution in and out of the pipette tip. After checking that the solution is homogeneously cloudy, I transfer 100 microliters of solution from the 10 to the 7th to the 10 to the 6th tube. I repeat the mixing process with the pipette, gently pushing solution in and out to make sure yeast cells are evenly distributed. We then transfer 100 microliters of solution from the 10 to the 6th to the 10 to the 5th tube, and so on and so forth until we get to the last tube. At this point, we are ready to start plating. You are going to write your initials, the date, section, strain, and UVR exposure time on each plate. We have a 10 to the 3rd control plate and 3 10 to the 4th experimental plates with varying levels of radiation exposure. Here I am doing 5, 10, and 15 seconds. We do a control plate so we can compare the mutagenesis and survival rates of different experimental strains to one that has been chemically unaltered. We're going to take the F200, the P200 micropipette and pipette 100 milliliters or microliters of 10 to the 3rd yeast solution onto the first plate. You're going to take a spreader, unwrap it without touching the spreading end, and gently use it to spread the solution onto the plate. A good technique is spreading while rotating the plate and not getting too close to the sides of the plate. Next, we do the same with 100 microliters of 10 to the 4th solution onto the 3 experimental plates. Here are our 4 plates, and they are ready to go into the yeasty bake for UV-induced mutagenesis. You should set the control plate aside, it does not go into the yeasty bake. Open the oven lid and slide one of the experimental plates in, then turn on the red switch for however many seconds of UV exposure you want. Repeat this process for each plate. Next, we put the plates into the lab incubator for 2 days at 37 degrees Celsius. We wait a couple days, and here are our mutagenized plates. I used a lighter pink colony of HB1, so I am looking for creamy white mutants. You are going to count your colonies and see how many are red or pink and how many are white. It looks like we have some white mutants. As mentioned before, HB1 has a mutation in the AD1 gene, which along with AD2 is one of the more downstream steps of the adenine biosynthesis pathway. If add 1 or add 2 is mutated, the yeast colony accumulates an intermediate that is converted into a red pigment, and so the yeast will be pink or red. Exposing HB1 to UV can induce mutagenesis in an upstream gene of the adenine biosynthesis pathway so that the IR red pigment intermediate is never produced. This means that the resulting colony, while still an add1 mutant, will be creamy white. For my 5 seconds of UVR plate, there are 169 pink colonies and 12 white colonies. For 10 seconds, there are 107 pink and 12 white colonies, and for 15 seconds, there are 16 white and 18 pink colonies. We can see here that as time of radiation increased, the proportion of mutagenesis increased and survival rate decreased. These two are trade-offs for each other in terms of data and proportion, so watch out for that. You can use the data to plot a survival curve and do a repeat mutagenesis experiment with the calculated lethal dosages LD50 and LD80, which determine the rate of mutagenesis at which 50% and 20% of colonies respectively will survive. Thank you for watching.